All right, so here's pyruvate oxidation again up here. So here's our 3-carbon pyruvate. 1 carbon is going to be lost during pyruvate oxidation because 1 carbon gets oxidized, NAD plus gets reduced because we always have both oxidation and reduction. So here's our 2-carbon bit here, our acetyl-CoA. Acetyl-CoA is another important metabolic intermediate. Acetyl-CoA can leave at this point and go to making fatty acids. That's why fatty acids always have an even number of carbons, 8, 12, 18, 22, because they are synthesized from acetyl-CoAs being linked together, which have 2. Fatty acids, when they're broken down for energy, also can enter at this point. So fatty acids are broken down into 2 carbon bits, and those are bonded to a coenzyme A and turned into acetyl-CoA, and they can enter the cycle at this point. So in addition to G3P, acetyl-CoA is another metabolic intermediate. This is another important one also for amino acid synthesis. So amino acids can be broken down into acetyl-CoA. Acetyl-CoA can also leave and go to amino acid synthesis. Okay, so now if acetyl-CoA is going to enter the citric acid cycle, it's called that because the first molecule that acetyl-CoA binds to, notice it loses the enzyme CoA, that first molecule that forms is citric acid, so that's why it's called the citric acid cycle. So during the citric acid cycle, so this is the total outputs here of the citric acid cycle. We're going to lose 2 carbon dioxides for each acetyl-CoA that enters the cycle here. We're going to, because this is oxidation, there's some reduction. So we're going to generate some reduced NADH. We are also going to generate some FADH2. So remember, FAD is another electron carrier just like NAD+, but it can grab electrons that are lower in energy, but still have some energy. They just enter the electron transport chain at a lower energy level. And then we're going to make a small amount of ATP also. So here's all the intermediates. And again, each blue circle is an enzymatic reaction. So it's an 8-step process here. And some of these steps are oxidation reduction reactions. So this one is because we're reducing NAD, so that means this molecule here gets oxidized. This one as well is an oxidation reduction reaction. 1 carbon gets oxidized and NAD plus gets reduced. Here's a phosphorylation step, so you notice GTP is the first product here. So GTP is used by quite a few cellular systems, including the ribosomes use GTP for protein synthesis. So this would be the source of that. It can also be turned into ATP at a small loss of energy. So here's another oxidation reduction reaction, and I know that because FAD is being reduced to FADH2. So if we look carefully, we can figure out which carbon is becoming oxidized here. So we have another oxidation reduction reaction here, and then we are ready to regenerate our starting molecule, citrate. So again, I've taken out the names of all the intermediates and all of the enzymes. You don't need to know those, but you should know what goes in and what goes out of the whole process here. So the inputs are acetyl-CoA, the outputs, a couple more ATP, lots of high energy electrons, mostly reduced NADH, but also some reduced FADH2 and carbon dioxide. So this is where 2/3 of the carbon dioxide that we exhale comes from this step. And now all the carbons are fully oxidized. There is no more energy in this carbon. In pyruvate, we still have quite a bit of potential energy there. Acetyl-CoA, still quite a bit of potential energy left in that molecule. Once we finish the citric acid cycle here, there's no more energy in the CO2. Now the energy is all found in ATP and in these high energy electrons, which are ready to go on to the last step here, oxidative phosphorylation. The step that finally we're going to use the oxygen that we inhale, and phosphorylation is referring to turning ADP into ATP. So here's the electron transport chain, but on a graph showing how much energy the electrons have. So here is the electron transport chain here, showing that as these electrons get passed from electron carrier to electron carrier, they're going to step down in energy at each step of the process. And you'll notice that here's the NADH electrons entering here. The FADH2 electrons enter a couple of steps down. They're a little bit lower energy. So as these electrons are passed down and lose energy at every step, the work that's being done with this energy is turning one type of potential energy into another type of potential energy. And that energy, we are going from the energy of high energy electrons to the energy of a gradient. If you have a gradient across a membrane of an ion, like a sodium ion gradient, or in this case, it's going to be hydrogen ions, which we could also just call protons, because that's all they are. If you have a lot of protons on one side of the membrane and fewer on the other side of the membrane, that is potential energy. That's like having water behind a dam. You could allow those protons to flow across the membrane, down their concentration gradient, and that's going to generate energy. Okay, so here's our oxygen down here. Water is the lowest energy here. There's no stored energy in water, but it's oxygen that's the electron acceptor. So the electrons are drawn down the electron transport chain because of oxygen, because oxygen is sucking the electrons from the end of the electron transport chain, and then they end up with water. So that's what makes it flow. And again, here's a YouTube video that I have a link to in the PowerPoint that you all have. And I will also put this same video in the playlist for this section. So you can watch that after the end of this, because this is just another YouTube video here. So here's a close-up of the inner membrane of the mitochondria. So this is that membrane of the ancient bacteria. That's where the electron transport chain is. It's in the membrane of that ancient endosymbiotic bacteria. So up here, the orange, this is the intermembrane space. So this is the space in between that ancient bacterial membrane and the eukaryotic membrane. And then down here, this is the cytoplasm of the ancient bacteria, the mitochondrial matrix. So electron transport chain, and then, so the work that's being done here by the electron transport chain is pumping hydrogen ions, which are protons, across the membrane. So this intermembrane space will have more hydrogen ions than the matrix of the mitochondria. So think about what that's going to do to the pH of that space. If you add extra hydrogen ions to a solution, what does that do to the pH of the solution? It makes it more acidic. So this intermembrane space, if the mitochondria are functional, is going to be a little bit acidic relative to the matrix of the mitochondria or the cytoplasm of the cell. And then the last step here is the enzyme that's going to phosphorylate ADP. And that's when I've left the name of this enzyme on here because I would like you to know the name of this enzyme. That's ATP synthase. And we can see that ASE on the end that indicates that this is an enzyme. So ATP synthase allows these protons to flow down their concentration gradient, and it uses that potential energy to phosphorylate ADP. So, and again, I have this is another YouTube video here that I will put in the playlist right after this video so that you can watch that instead. If you have the PowerPoint, you have the link right embedded into the PowerPoint, but I'll put it on this playlist as well so that you have it. Okay, so here's ATP synthase and the intermembrane space. We have a concentration of hydrogen ions. That's a form of potential energy. We have the energy found in bonds, and that's electron energy. We have those electrons being transferred through the electron transport chain, so still electron energy. Now that potential energy is turned into the energy of an ion gradient across a membrane. That's another kind of potential energy that cells use. So the hydrogen ions flow down their concentration gradient through the ATP synthase molecule, which actually rotates like this. When this was discovered about 30 years ago, that's what this molecule actually did. It's weird and was very surprising to the scientists who discovered this. But it actually rotates. And as these hydrogen ions flow down their concentration gradient, they force the rotation of this molecule, and that energy is what is used to phosphorylate ADP. So we have ADP plus inorganic phosphate is turned into ATP, which has more potential energy than these together. So the energy goes from electrons to a proton gradient to ATP. So here's our summary of all of this. So we have glycolysis, which generates a little ATP, some high energy electrons, and then we have pyruvate oxidation, which generates some high energy electrons, no ATP though, and a carbon dioxide here. And then the citric acid cycle, lots and lots of electrons, NADH and FADH2, and 2 more carbon dioxides per pyruvate, and a little bit of ATP. And then finally, oxidative phosphorylation, electron transport chain, and chemiosmosis generates a significantly higher amount of ATP. And also, all of these electron carriers are going to be oxidized. So when they react with the electron transport chain, they are oxidized. When they give up their high energy electrons, they are oxidized. So oxidation is losing. They lose those high energy electrons. And maximum here, total ATP per 1 glucose, 30 or 32, but really most cells don't get that much. Okay, so fermentation is when glycolysis alone is going to be used, like in yeast or in your own muscles when you're doing anaerobic activity, like sprints or lifting really heavy weights. Imagine something that's intense enough that you're not going to be able to continue it for more than a few minutes. That's anaerobic exercise. So, and in that case, then your muscles want to continue glycolysis because the mitochondria can't keep up. There won't be enough oxygen to generate a hugely increased amount of ATP. So, but there's a problem. The reason why glycolysis alone cannot just continue is because we're going to run out of oxidized NAD+. And if you have an oxidation reduction reaction, you have to have both components. So if we just have glycolysis alone here running, eventually we're going to run out of oxidized NAD. And now glycolysis is going to screech to a halt. So what the fermentation reaction does is it allows glycolysis to continue by wasting these electrons. So these high energy electrons, instead of going into the mitochondria to make ATP, they are going to be wasted, and that's going to allow glycolysis to continue. So most cells do lactic acid fermentation, and that's prokaryotes and eukaryotes. This is what our cells do, lactic acid fermentation. So glycolysis, we generate pyruvate. If the pyruvate is not going to move into the mitochondria for oxidative respiration, which this would be in a case where you were doing intense short-term exercise in your muscles, then pyruvate is going to be reduced to lactate. And NADH is going to be oxidized to NAD+. Now, there's still a lot of energy in this lactate molecule, and it can be used by mitochondria. Lactate can be used by mitochondria just like they use pyruvate. So what happens when you're done exercising, you have a lot of lactate built up in your muscles, and the lactate will then be slowly reabsorbed by the mitochondria in your muscle cells. If that doesn't get rid of all of it, it will end up in the bloodstream, it will go to the liver, and then your liver will deal with it. But all this energy is eventually going to be used in the mitochondria. Yeast do something weird. Yeast are single-celled fungus, and yeast are competing with a particular other kind of organism in their environment. So yeast live in sugar-rich environments like rotting plant material, rotting fruit, and they're competing with bacteria in that environment. Bacteria can divide much more rapidly than yeast. Yeast are a eukaryotic cell. It takes them an hour and a half to 2 hours to divide. Bacteria can divide in 15 or 20 minutes. So what they have evolved, because they've been competing with the bacteria for about 2 billion years, so what they have evolved is a type of fermentation reaction that produces a poison. They produce ethanol as a byproduct, and the yeast cells can tolerate a much higher alcohol concentration than most bacteria can. So bacteria divide really rapidly. Yeast have evolved to poison them so that they can have the glucose. Now, of course, there are bacteria that can survive that, but most bacteria are going to be poisoned by that. So yeast fermentation produces carbon dioxide and ethanol, which is a 2-carbon alcohol. But it accomplishes the exact same thing as lactic acid fermentation. Reoxidizes NAD so that glycolysis can continue to produce its small amount of ATP. And again, here's another embedded video. This one is not a YouTube link, but I'm going to try to find a similar one to put in the playlist so that you can watch that. Lactic acid fermentation is used in the food industry. If you're consuming any kind of a soured dairy product like yogurt or sour cream or cheese, very often it's a bacteria that's doing lactic acid fermentation that's giving it that sour taste. So very useful. Okay, so glucose is broken down to pyruvate, and then one of two things can happen. If it goes into the mitochondria and it continues for oxidative respiration, we're going to get a lot more ATP. But if there's not enough oxygen present, or if it's an organism like yeast that it doesn't matter to the yeast if there's oxygen present, it would still prefer to ferment even if there's oxygen around. Then we have fermentation, which produces far fewer ATPs, but generates energy without using oxygen. And as we'll see when we get to mitosis and we talk about cancer cells and cell division, cancer cells also use this pathway and lactic acid fermentation and produce far fewer ATPs and are therefore very glucose hungry. But we'll talk more about that in a later chapter. Glycolysis is probably the oldest type of metabolism. Almost all cells can use or make glucose and use some version of glycolysis in their metabolism, which kind of makes sense because, as we talked about for the origin of life, there wasn't any oxygen available on the early Earth, and even when oxygen started to become free oxygen started to be available after cyanobacteria first evolved, it was still very low amount of oxygen for a long time. So glycolysis would have been much more useful. aerobic metabolism could not have evolved until there was a substantial amount of oxygen in the atmosphere, and we don't think it evolved until there was about 2% of the atmosphere was oxygen. So that did not happen until about 2.5 billion years ago. What about all the other food that we eat? So you probably know that we can use fat and protein for energy as well. In fact, we do every day. If you eat more protein than your body needs, strictly speaking, to rebuild all the tissues, then some of those amino acids are being broken down for energy. And fatty acids are also broken down for energy. And fatty acids enter directly as acetyl-CoA. Fatty acids have an even number of carbons. They're broken down into acetyl-CoA, and they enter directly into the citric acid cycle. Amino acids vary in size quite a bit. If you remember the structure of amino acids, the smallest amino acid, glycine, only has 2 carbons. Other amino acids can be broken down into 3 or 4 carbon units. And so they're going to enter either the citric acid cycle directly, or a 2-carbon unit might enter as acetyl-CoA. So carbohydrates enter as sugar. Fats, when we break down a triglyceride into a glycerol and 3 fatty acids, the glycerol can enter as G3P. Fatty acids will be broken down into 2 carbon bits, and these can be reversed as well. So if we're going to be storing fat, glyceraldehyde 3P will form the glycerol backbone, and acetyl-CoAs will be linked together into fatty acids. And for amino acids as well. It depends on how many carbons they are broken down into. If it's a 3-carbon bit, then it will enter as pyruvate. If it's a 2-carbon bit, acetyl-CoA. If it's 4-carbon bit, citric acid cycle directly. And of course, that amino group that's on many, there's at least one amino group on every amino acid, but there's also amino groups in the side chains as well. Though that has no stored energy, because just like oxygen, nitrogen is an electronegative electron hog. So things bound to nitrogen don't have any stored energy. So amino groups in ammonia don't have any stored electron energy. So if we're burning amino acids for energy, we just have to get rid of this in waste, and these amino groups end up in the molecule urea and end up being excreted in our urine. So feedback, negative feedback and positive feedback. Feedback mechanisms change a reaction. So if it's a negative feedback on a reaction, that means it's going to slow it down. The feedback tends to reduce the amount of the reaction that's happening. Positive feedback would mean that the products of the reaction are going to actually increase the rate of the reaction, and they're less common, but they do exist. So for example, here is one of the enzymes in the middle of that glycolysis pathway, one of the enzymes that I took out of the original diagram there, phosphofructokinase. And this is a common occurrence in metabolic pathways. Not all the enzymes in the pathway will be regulated, because you really only have to regulate one enzyme near the beginning of the pathway. And what that will do is prevent all the other enzymes from having any substrates. So you don't need to regulate everyone. And remember, inhibit means stop. So when an enzyme is inhibited, that means it's going to lower the number of reactions it's going to catalyze, or completely stop it. If it's stimulated, that means it's going to increase the number of reactions per second that this particular enzyme is catalyzing. So phosphofructokinase is near the beginning of glycolysis. So we can either inhibit or stimulate that. So citrate or citric acid negatively feeds back on phosphofructokinase and slows it down, because if we have an excess of citrate around leaking out into the cytoplasm, that indicates that there's plenty of products of the citric acid cycle around, and we should probably slow down glycolysis. As well, lots of ATP in the cytoplasm does the same thing. It slows down glycolysis. So the amount of ATP is in the cytoplasm of a cell is pretty tightly regulated. As ATP goes down, eventually AMP is going to be formed, adenosine monophosphate. This indicates the cell really needs more energy right now. So AMP binds to phosphofructokinase and increases how many reactions per second that it's going to catalyze, because we need more ATP if there's lots of AMP around. Okay, so ATP is the molecule that's used to store energy in the cell. GTP is secondarily used. So ATP is used for muscle contraction and all sorts of synthesis reactions. GTP is used for protein synthesis and G proteins and a few other specific things. Almost all eukaryotes and many prokaryotes as well use this type of aerobic cellular respiration to make ATP. All eukaryotes either have mitochondria or have dysfunctional reduced mitochondria that they don't use anymore. Glycolysis is in the cytoplasm. Everything else takes place in the mitochondria. We talked about the Krebs cycle or the citric acid cycle, electron transport chain, and I will link to those videos that are in the PowerPoint so that they'll just be in the playlist that goes along with this. So look for those. Bye. That's the end of respiration and fermentation.